The human language likely emerged millions of years ago, but there is no record of the first word, song, prayer, or even the first dialogue.
Language gave us the ability to escape the present reality and plan for the future, to imagine other worlds. It is language that creates cohesive societies, organizing beliefs, behaviors, customs, and social values. Verbal language is our species' superpower. Speaking for humans is like flying for birds. It occurs naturally, but it is the result of millions of years of evolution, which has fostered a complex interaction between muscles and the brain.
And because it's complex, it's also very fragile. A single point mutation in our DNA, targeting a gene such as FOXP2 or any other autism-related genes, can significantly impact language. But how exactly did language arise in the first place? Could our extinct relatives, such as the Neanderthals, could they speak?
In collaboration with our international colleagues, we have found that ancient hominids, including early humans and Neanderthals, were exposed to lead much earlier than in modern times. It's long been assumed that humans have been exposed to harmful amounts of lead since antiquity, when the Romans used lead pipes to transport water, and that lead contamination increased significantly during the Industrial Revolution, only to be curtailed during the late 20th century. We stopped using lead in our daily lives when we realized how toxic it is, but nobody had ever studied lead in prehistory. This was concluded after the analysis of fossilized teeth across Africa, Asia, and Europe. Surprisingly, teeth from people born between the 1940s and 1970s, when children were exposed to leaded gasoline and paint, showed similar patterns of lead exposure to fossilized human teeth.
We hypothesized that, like the Romans, ancient humans and other hominids might have been exposed to lead due to their needs for water. One possibility is that they were looking for caves with running water inside. Caves contain lead, so they were all contaminated. Based on the tooth enamel studies, it started very early in infancy.
Lead exposure impedes brain development, leading to deficits in intelligence and difficulties with emotional regulation. Given these findings, Muotri and his team wondered how the modern human brain had flourished despite exposure to lead during our evolution.
A gene called NOVA1 plays a central role in neurodevelopment. It affects neuronal maturation and synaptogenesis. Alterations in the activity of NOVA1 is linked to several neurological conditions.
Most modern humans have a variant of NOVA1 gene that differs by single DNA base pair from the ancestral version that was present in Neanderthals. Previous work by Muotri and his colleagues demonstrated that replacing the human NOVA1 variant with the archaic variant led to significant changes in the architecture and synaptic connectivity of tiny stem cell-derived models of the human brain, known as brain organoids. The archaic variant accelerated brain maturation, but resulted in less complexity over time. If all humans have this newer mutation in all corners of the world, very strong genetic pressure must have selected for it in our species.
Interesting, NOVA1 also controls the lead response in neuroprogenitor cells. To understand if lead contamination from the environment participates in this selection, we expose brain organoids carrying either the archaic or the modern version of NOVA1 to lead, and we analyze the consequences using cortical and thalamic neurons.
Lead exposure altered NOVA1 expression in both variants, affecting genes linked to neurodevelopmental disorders such as autism and epilepsy. However, only the archaic NOVA1 variant changed the expression of FOXP2, a gene essential for language and speech development. People with FOXP2 mutations are unable to produce sophisticated language. These type of neurons related to complex language are susceptible to death in the archaic version of NOVA1. The FOXP2 gene is identical between us and the Neanderthals, but it's how the gene is regulated by NOVA1 that likely contributes to language differences. The findings indicate that acquiring the modern NOVA1 variant may have shielded us from the harmful effects of lead, fostering complex language development and social bonding. This could have provided modern humans with a significant evolutionary edge over Neanderthals, even when exposed to lead contamination. More recently, an independent study using a NOVA1 humanized mouse model showed altered vocalization, adding evidence that NOVA1 might have contributed to the development of spoken language in modern humans.
Our work produced evidence that the human brain is an outlier in terms of complexity and language. Not even our closest relatives, the Neanderthals, might have the cognitive capability to produce sophisticated language.
Understanding how NOVA1 influences FOXP2 expression helps to clarify the connection between lead contamination and brain development, and will contribute to the understanding of neurological conditions related to language, including speech apraxia and autism.